Just wait for a couple more folks to join. Excellent. Good morning or afternoon, everyone, depending on where you're calling in from. Welcome to today's webinar titled Sleep, Circadian Rhythms and Health, Looking Forward to Healthy Aging. I'm Dale Sampson, VP of Bioinformatics at Elysium Health, and your host for this session. Today, we're honored to have Professor Russell Foster join us. Professor Foster is the head of Oxford's Nuffield Laboratory of Ophthalmology, the founder and director of the Sleep and Circadian Research Institute, and a fellow of Brasenose College, Oxford. He also serves as a valued team member, as a valued Elysium Health Elysium Scientific Advisory Board member, and a principal investigator for Elysium's sleep study titled Time ZZZ. Professor Foster's research focuses on generation and regulation of circadian rhythms and sleep, particularly addressing their disruption due to societal pressures, aging, and disease. He's renowned for his discovery of an unrecognized light detecting system within the eye that regulates these rhythms and sleep, and for translating these findings into clinical applications. His accolades include election to the Fellowship of the Royal Society, the Royal Society of Biology, and the Academy of Medical Sciences. In 2015, he was appointed a Commander of the British Empire for his services to science. Professor Foster has authored over 300 peer-reviewed scientific papers, received numerous national and international awards, including the Daylight Prize, and has co-authored several popular science books. Currently, he's working on a new book exploring the role of light in the biology of humans, animals, and plants. With such an illustrious career and profound contributions to the field, we're eager to hear his insights today. During today's webinar, we won't be taking live questions, but I urge you to ask any questions that you have and put them into the chat window. We're going to collect these at the end and compile them and send them back out with responses in an upcoming newsletter. So without further ado, it's my absolute pleasure to introduce to you Professor Russell Foster.
Thank you very much indeed, Dale. I'm really delighted to join you today. I have to say, you're looking remarkably perky from flying back from Australia. You must have appalling jet lag. So congratulations.
I made it.
So as Dale was saying, I want to touch on sleep, circadian rhythms, and health. And again, taking an optimistic look forward to the future and healthy aging. I thought I'd spend our 60 minutes together by considering, first of all, the biology of circadian rhythms and sleep. So a gentle introduction to the topic. Then talk about the importance of biological time. What we do when really does matter. Then circadian rhythms, sleep, and the impact of SCARD, and it's a term we've introduced, which has now been broadly adapted, which is an abbreviation for sleep and circadian rhythm disruption. Then talk about, and really the next 3 bullet points are all about this translation initiative to map epigenetics in sleep, the Time ZZZ, or should I say ZZZ program. So I'm introducing DNA methylation, circadian rhythms, and age. Then some of the work that we're doing, which kind of overlaps, and the translation from fundamental knowledge into the development of new drugs. And then for the last few slides, outline the project, which I hope many of you will be able to join. So if we kick off with the biology of circadian rhythms and sleep, and to state the glaringly obvious, we sit on a planet that revolves once every 24 hours. And of course, that results in immense complexity for life on Earth. We have the day and the night. So light, dark, food availability, social interactions, predation, non-predation, completely different environmental realms. And essentially all life on the planet, very much including us, has evolved specializations to deal with the day and the night. So in our case, physical inactivity during sleep. But as we'll see, sleep is a very, very important biological function. It's not just simple physical inactivity, a lot going on in the brain whilst we sleep. And of course, physical activity, consciousness, going about all the stuff that we need to do. And critically, these two systems, the sleep and the consciousness systems, are constantly interacting. So the quality of our sleep will define the quality of our consciousness and vice versa. So I'd like to make the point that our body clock or circadian system anticipates this 24-hour light-dark cycle and adjusts and in fact fine-tunes our physiology in advance of the changing conditions. So it prepares us for the challenges ahead. And as a result, our biology is highly dynamic. And let's look at a few examples. So you see 6 boxes here, and each has a 24-hour time base. And the gray area here is sort of representing night. And the first rhythmic change I want to show you is melatonin, a hormone that many of you will be familiar with. I would stress that it is emphatically not a sleep hormone. It's a biological marker of the dark. And please send me questions about melatonin. We can answer those later on. And the key point I want to show you about melatonin changes under the influence of the clock is that they rise in anticipation of the nighttime, and they drop while it's still dark in anticipation of the dawn. And this is a classic example of anticipation. And that's what we see in essentially all of our biology and within the examples I'll show you now. So for example, growth hormone is almost exclusively released during the first half of the night. Interestingly, there's a rhythm, a circadian rhythm in our core body temperature, and it drops towards evening. And in fact, that drop in core body temperature is probably an important signal for sleep initiation. Cortisol rises in anticipation of activity, mobilizing glucose, increasing heart rate for the increased demands of activity. An interesting one here, testosterone rises in the morning, and that's been intriguingly associated with increased sperm motility at this time of day. And as we'll see later on, multiplication accuracy and cognition generally drops to a low point in the early hours of the morning and then rises to around about its peak point at noon. Okay. So these are the dynamic changes that you see in much of our physiology. And the key point again is that the circadian system delivers the right materials, the right stuff in the correct concentration to the right tissues and organs at the correct time of day. It's absolutely essential. Essentially, without this time structure provided by the circadian system, our biology, including our sleep, of course, would be plunged into chaos. Okay, let's consider the generation of circadian rhythms. And for that, we'll go into the brain, and we see the human brain in sideline profile down the middle, and we see this tiny structure here. This is the optic nerve. So as the optic nerves come into the brain and fuse, we see this basal bit of the hypothalamus. And if we enlarge that bit here, you can see this paired structure within the hypothalamus. And here's a cartoon of it called the suprachiasmatic nuclei, or the SCN. And you see this is the optic chiasm where the optic nerves come in and fuse, and we've got this extraordinary paired structure here. It's about 50,000 cells. And what's, I think, truly extraordinary, one of the great success stories of neuroscience in general, is the discovery that each one of those cells in the SCN can produce clock genes, and those clock genes produce clock proteins, but those clock proteins feed back and turn off those genes. And I'll go into the molecular clock in more detail towards the end of the presentation. The key point is you've got this molecular oscillation of clock proteins. And this is the essential signal that's coming out of the master clock within the brain, the suprachiasmatic nuclei. Now, it's no good having this wonderful coordinator, this coordinator of our rhythmic activity, unless the internal day is set to the external world. The clock has to be entrained, and the primary agent that entrains the clock is the light-dark cycle. And that light-dark cycle is detected by the eye and the retina in the eye. And as you see over here, and we'll talk a little bit more about the retina in a minute, and you see that there's a direct projection from the eye called the retinohypothalamic tract, which sets the clock cells to the external world. And that's absolutely critical. And I will touch on this again briefly in a moment. Okay, well, those molecular signals need to be turned into a physiological response. So what I've illustrated here is the electrical activity within the SCN during the day, when there's few spikes, a low firing frequency, compared to the night is here, and during the day you have a high firing frequency, lots and lots of electrical activity within the SCN. Now, what's turned out, I think, to be absolutely fascinating, that this pattern of electrical activity, and indeed the molecular changes you see in the SCN, are the same for a day-active animal such as ourselves and a night-active animal such as a mouse. So the decision to become nocturnal is not at the level of the SCN, but it's actually downstream. And we don't understand actually how nocturnal versus diurnal behavior arises. One of the other great discoveries over the past few years is that these clock cells that we see in the suprachiasmatic nuclei, and we originally thought there were only clock cells in the SCN, but now we know they're found basically in every cell of the body. So what's happening is that the SCN is coordinating literally billions and billions of individual cellular clocks throughout the entire body, which is coordinating that rhythmic activity that I showed you earlier. So what we have is not, and we often use the shorthand, the biological clock. Actually, it's a circadian system, a massive network of biological time. Okay, I said I'd come back to light. Here we go. Just to emphasize what I said earlier, under a light-dark cycle, as you see illustrated in this cartoon here, the peak of rhythmic activity of the clock occurs at the same time every day. But if I take an individual and we put them under constant darkness, or tragically, if you have no eyes, then the clock will still tick, but you see that it gets later and later in time. That's true for about 90% of us. Some of us have short clocks, but most of us, 90%, will have a long clock. So we'll get up later and later and later each day. And it varies, 11 minutes, 10 minutes, or half an hour, depending upon the individual. So the key question that we asked quite some time ago now is, how is the SCN entrained by light? Here we see the eye. We need an eye, and here's a cartoon picture of the retina. Now these are the visual cells, the rod and cone photoreceptors, and the rods give us our nighttime dim light vision, and the cones give us our color vision during the day. This is the inner retina, where signals from the rods and cones are integrated before messages are sent via these ganglion cells and the optic nerve into the brain. And the assumption, of course, quite rightly, was that it's the rods and cones detecting the light-dark to regulate the clock. We tested this assumption quite some time ago now, first of all using mice with hereditary retinal disorders where the rods and cones has degenerated, and then later some transgenic animals in which the rods and cones had been completely turned off. And so what we found is, with no rods or cones, the mice could entrain their biological clock perfectly normally. So you could be visually blind, but not clock blind. We then demonstrated that humans also had something else in the eye. They didn't need their rods and cones to detect the light-dark cycle. And finally, that led to work by us in the mouse, Dennis Dacey in the monkey, and others on the rat, and showed that about 1% of those retinal ganglion cells are directly light sensitive using a photopigment, a light sensitive molecule that was called melanopsin, or OPN4. And this OPN4 is based in these cells, which we've called photosensitive retinal ganglion cells. So it's worth just reflecting that the eye is the organ, of course, organ of space. It gives us our vision, but it's also the organ of time because of its ability to regulate the internal clock. And these dual functions of the eye are now being studied in ophthalmology departments all over the world. We're doing quite a bit of work on trying to assess which ocular diseases will impact upon these cells and the ability to regulate the circadian system. So quite literally, this is I suppose, classifying our understanding of what we mean by blindness. This is what they look like. They're just beautiful, forming this photosensitive net across the arc of the eye. And I can't resist showing you a high-power picture of these cells. They are just beautiful. So anyway, bear with me. I think they're perhaps the loveliest cells on the planet, but there we go. But I've talked about the circadian system and its regulation by light, but we also need to think about sleep because it's more than just the clock saying go to sleep and get up. If we again look into the brain, we see that there are two powerful systems. There's the wake-promoting systems, one of which is in the lateral hypothalamus, producing orexin or hypocretin, which is promoting the release of histamine, dopamine, noradrenaline, serotonin, acetylcholine, glutamate, these neurotransmitters that are bathing our cortex, our brain, and giving us our sense of consciousness. And that's essentially opposed by the sleep promoting systems, which effectively, but not completely, turn off those wake promoting systems. So we have galanin, an inhibitory neurotransmitter in the ventrolateral preoptic nuclei, and of course big GABAergic systems which act to inhibit the release of those key neurotransmitters, except at one point acetylcholine is actually turned on during REM sleep. And you'll be familiar with the concepts of non-REM sleep and rapid eye movement sleep, and this flip-flop between the two states every 70 to 90 minutes or so, regulated in the midbrain. And of course, during REM sleep, we have our most vivid and complicated dreams. And whilst we're having those dreams, we exhibit atonia or muscular paralysis. So you've got this amazingly complicated set of interactions between all of the key brain neurotransmitter systems. And I'll come back to this point when we talk about mental health. But what flips between the wake and sleep states? Well, there's the circadian drive, and then there's the sleep need, or the homeostatic drive for sleep. And let me just explain how those two systems interact to generate the sleep-wake cycle. And this is a concept introduced really quite some time ago, the two-process model by Alex Borbley and Sergey Dan. I knew Sergey very well, tragically died in 2018. Wonderful man. So the first point I want to look at is the sleep pressure whilst we're awake. So from the first point in the morning, and as we go through the day, our drive for sleep increases and increases and increases. And then as we sleep, this sleep pressure, or this is dissipated. Now, the key point is what stops us from falling asleep when the sleep pressure here is really high. And for that, we need to look at the important circadian drive for wakefulness. So we see a low drive for wakefulness in the morning because the drive for sleep is low. As the sleep pressure increases throughout the day, the circadian drive for wakefulness keeps up with it. If it doesn't, this is why we may want to have a mid-afternoon sleep, where the sleep pressure and the circadian drive have been slightly uncoupled. Key point is the circadian drive for wakefulness is at its highest just before we go to sleep. Then the clock will say, okay, it should be sleep time. So the wakefulness drive diminishes, the sleep pressure kicks in, you fall asleep. And then, of course, whilst we are asleep, the wakefulness drive from the clock is low. And then as we approach morning, the wakefulness drive is increasing it exceeds the tiny low levels of sleep pressure, and at this point you wake up. And so it's these interacting systems that coordinate our rest activity, our sleep consciousness cycle. And just to, again, to emphasize, whilst we're asleep, it's not that the system is shut down, as we'll discuss, but it's a mass of essential biology that occurs during this time. Okay, I hope that's given you a flavor for the biology of circadian rhythms and sleep. Now let's look at the importance of biological time. As I said, what we do when really does matter. And the concept I want to start with is the concept of chronotype. And I suspect many of you will have heard of the morning versus evening types. So I thought we'd dig into that a little bit in a bit more detail. So this is a distribution of chronotypes across the population. And what we see here is the doves, which is most of us, 65% of us are somewhere in the middle. We're neither larks, extreme morning types, and that's about 10% of the population, or owls, which are late types, which is about 25% of the population. Now, there's a real biological basis for whether you're a morning type or an evening type, or somewhere in the middle. And I thought I'd talk about the basis for chronotype in a bit more detail. And there's basically three things that we need to take into consideration. First of all, genetics. So we know that key genes, tiny changes in some of those clock genes, can either nudge you into being a morning type or an evening type. So by their contribution to our genes, our parents are still telling us when to go to bed and when to get up. But also, it's likely that there will be epigenetic influences on our chronotype. And this is one of the things that we'll be exploring in the project, the Elysium project, the Time ZZZ trial. And I'll come on to that towards right at the end of the presentation. So there's genetics and probably epigenetics. But when you see light is immensely important. So morning light advances the clock. So when you wake up in the morning, you see light, the clock will make you, will be adjusted earlier in time. You'll want to get up earlier and go to bed earlier. And in the evening, there's the opposite effect. So dusk light delays the clock. So you want to go to bed later and get up later. Now, when we were all agricultural workers, we got a symmetrical exposure to the light-dark cycle, the morning-evening cycle, and we stayed on cue. One of the great problems at the moment is that exposure can be asymmetrical. And we did a study on university students all over the world a few years ago showing that the greater, the more you were an owl, an evening type, the more evening light you got, delaying the clock compared to morning light, which would advance the clock. So evening types would sleep for the morning, miss the morning light, but they'd be out in the late afternoon, early evening, get the evening light, which would delay the clock. So when you see light, morning versus evening can be incredibly important. But a third factor is age. And in fact, there's an important difference between males and females. So let me show you those data. So here we have age on this axis, going from 10 years to 80 years. And here's chronotype from morning to evening on this axis. And what you see here is that from around about the age of 10, there's a tendency to want to go to bed later and later and later. Females peak at around about 19 and a half. Males peak later at around about 21 and a half years of age, and males tend to be later chronotypes than females. And then as we age, we go back to being more morning types. So by the time we're in our late 50s, early 60s, we're getting up and going to bed at about the time we got up and went to bed at the age of 10 or so. But the key point is that there's about a 2-hour biological difference in preference between bedtimes compared to somebody in their late teens, early 20s, and somebody in their late 50s, early 60s. And it seems that this change is driven by the hormonal changes associated with puberty. So increased levels of testosterone in males, estrogen and progesterone in females. And we have now good studies from our lab and all over the world showing how these sex hormones interact with the molecular clockwork. Okay, so that deals with chronotype. Let's now look at the impact, oh yes, I should say that in teenagers, of course, chronotype, the biological predisposition to go to bed late and get up late, is exaggerated by social media, gaming, and all the other stuff that you can do online. But I want to have a look at an application of chronotype, just to illustrate how chronotype can be important. And this is on performance, and specifically athletic performance. So this is a nice study that came out in 2015 from the University of Warwick, looking at a running task. The details are unimportant. But what was extraordinary is that for the early chronotypes, their peak performance was around about 12 o'clock noon. For the intermediate chronotypes, around about 3 o'clock in the afternoon. But the late chronotypes were around about 7 o'clock in the evening. So you see here a real biological effect of pushing one's physiology to the extreme and the impact upon chronotype. Of course, this has big impacts upon sports. Most of the sports, the track and field events, tend to be in the late afternoon, early evening, which would favor late chronotypes, for example. Okay, and I couldn't resist this because genius that Benjamin Franklin was, I do feel that he did a disservice by saying early to bed and early to rise makes a man healthy, wealthy, and wise. Well, there's lots of things there that I would object to. My experience, the difference between a morning type and an evening type is that people that get up in the morning are just irredeemably smug. And you know, there was a paper published last week which kind of supports that view. This was a lovely paper, looked at 26,000 people. And the bottom line is that those who stay up late and those classed as intermediate, so the doves and the owls, had superior cognitive function, while morning larks had the lowest scores. I find that quite extraordinary. But then I'm biased because I'm a late type. Going to bed late is strongly associated with creative types, such as artists, authors, musicians, and I would throw in there scientists. And finally, sleep duration. This is important. Sleep duration is important for brain function, with those getting between 7 and 9 hours of sleep each night perform best in cognitive tests. So no matter what chronotype you are, as long as you're getting your sleep duration, you're doing okay. I think this is also important because it shows an awful lot of variation in terms of sleep. One shoe size does not fit all, and that's the same for sleep. Some people can get away perfectly well with 6 hours of sleep. Others may need 10 hours of sleep. And what we all have to do is assess what each of us needs in terms of our sleep needs. I want to talk about cognition now, our ability to process information. So this is a study from Christian Hjocken's group in Switzerland. And we see the time base here is from 7 a.m. to 11 p.m. And what Christian looked at was a whole range of different cognitive tasks, our ability to multiply, our speed, reaction, drawing accuracy, problem solving, memory recall, and showed that in adults, you have a peak in cognitive abilities at around about noon. Now, that will vary, of course, depending upon chronotype, but that's the average. And so people will argue that if you have important decisions to make, an important sort of task to perform as an adult, then you should do it around about lunchtime. But as I said, teenagers and young adults are different. They're about 2 hours delayed. And I think this represents actually quite an interesting dilemma in education, because, of course, traditionally, most of the so-called demanding subjects like maths and the sciences are delivered first thing in the morning. And so, yes, that's great for the teachers, the adults. They're coming in and they're feeling, you know, peak of their cognitive abilities, all great. But look at the kids, the teenagers are way down here. By the time the teenagers are showing peak cognitive performance, the teachers are looking decidedly jaded. So there's an interesting mismatch between teenagers and their teachers in cognitive performance. Incidentally, when exams have been moved from the morning to the afternoon, scores go up, which I think is something we need to take into consideration. We now want to extend it. Last time we talked about 7 a.m. to 11 p.m. I now want to go from 11 p.m. and add the rest of the day. And what you see is what we saw before, but after 11 p.m. cognition drops further and further and further. And I think we might all appreciate how low our cognitive abilities might be at 5 o'clock in the morning as a circadian driven change. But what Drew Dawson did from Australia was a lovely experiment comparing cognitive performance across the day versus the drop in cognitive performance when you were legally drunk. So here we have cognitive performance increasing on this axis. The low point in our cognitive performance is around about 4, 5 o'clock in the morning with a score of -20. So what is the drop in cognitive performance when you're legally drunk? It's -15. And so what this tells you, and perhaps it's the most important message in this whole talk, is if you're driving a car in the early hours of the morning, your ability to drive that car is worse than if you got into the car with sufficient alcohol to make you legally drunk, which of course explains why the accident rate accounting for traffic volume is the highest in these early hours of the morning. It's been estimated that there are 100,000 crashes on the American freeway due to these early hours of driving. Stroke. Okay, so if we look at the frequency of stroke, here's our blood pressure, and we see this very sharp rise first thing in the morning because we need to get blood to those tissues and organs that require higher energy demands. So you've got a sharp rise in blood pressure, but you've also, curiously, got an increase in plasma viscosity. The platelets that stick the blood together are most viscous at this time. Now, this isn't a problem under normal circumstances if you're healthy, but if you have hypertension, for example, then your chances of having a stroke are illustrated here. So what you get is that between 6 a.m. and 12 noon is almost a 50% greater chance of having a stroke than at any other time of the day. So when you look at your wristwatch and you see it's 1 minute past 12, you can have a massive sigh of relief because you know you've survived the most dangerous part of the day. This is well described, and our German friends call it, and I apologize for my accent, Todeszone, which is death zone. This is well recognized, but it raised some important questions. When to take your antihypertensive medication, morning or evening? Now, this was addressed by several groups, and I'll show you some data in a moment, but the issue was, okay, well, if this danger zone is between 6 a.m. and 12 noon, you've got up, you may start to take your antihypertensives, I don't know, 8 or 9 o'clock in the morning, then they've got to get into the blood and have an effect. And so in a sense, you're on the downward curve and might even have avoided completely the dangerous zone before your drugs can kick in. So what they did was over about a 10-year period, look at individuals and when they took their medication, their antihypertensive medication, and the incidence of death. This is bedtime treatment. They've taken the antihypertensives there, and this is the death rate. And very interestingly, if you take your antihypertensives after you've woken, then there's about a 50% difference in death. So it's now argued that taking your antihypertensives, assuming they have a long enough half-life, which is that they hang around in the body for a sufficient amount of time, you take them before you go to bed, rather than perhaps intuitively taking them after you wake up. cancer. We've known for a very long time that when you take when you're given your anti-cancer drugs can affect your long-term survival. And I'm showing you some deliberately older studies. So this is childhood leukemia. And so we've got weeks from diagnosis along this axis here, and the probability, essentially survival in complete remission. It's a relatively small study, and as I say, it's an early one, but this data has been around since 1993. And what you see is that the kids that took their or given their anti-cancer drugs, their chemotherapy in the evening versus the morning, you had 70% survival in the evening, 35% survival if the drugs were given in the morning. I mean, it's a huge difference. And this was a study over, what, 6.5 years. So same drug, same concentration, different time, big impact upon survival. And just to give you one more example, this is ovarian cancer. Again, same cocktail of drugs, but given at 2 different times. Schedule A, there was, after 5 years, about 45% survival. Schedule B, again, same drugs, different time, was reduced to 10% survival. And currently, this sort of information is not being integrated into our healthcare systems, and it's something that we may want to talk about later on. Immune function. So this is when you have your immunization against the influenza vaccine, and this is in individuals that were about 70 years of age, either in the morning, 9 o'clock to 11 o'clock, or the afternoon, 3 o'clock to 5 o'clock. And actually, again, remarkable results. So these are the morning results. These are the afternoon results. And so morning provided much greater protection. So the antibody response here was 400 with the morning versus 100 in the afternoon. A really remarkable finding. Whether this is also true for the COVID vaccines, we don't know yet, but those studies are being undertaken. But what it means and what these data are consistent with is that the adaptive immune responses, those that generate antibodies, are less robust when stimulated later in the day and in the evening compared to first thing during the day and throughout the midday. Okay. And finally, I just want to touch on when we do our drug testing. Now, quite rightly, we test our drugs on mice and then we extrapolate to humans, but often that extrapolation is not done appropriately. And drug testing is performed on nocturnal rodents and extrapolated to diurnal species. So this illustrates the different circadian patterns of a mouse and a human. And you see they're about 12 hours shifted. And this is a drug that was developed to improve the outcome of stroke and was tested on mice. And the researchers came in the morning, gave the drug, and it was extremely effective at reducing the damage caused by a surgically induced stroke. So this was then extrapolated to humans. So researchers went in, gave the drug at the beginning of the wake phase, and it wasn't effective. And then finally the penny dropped and they said, well, hang on, this is the beginning of the human wake phase. What happens if we give it at the beginning of the mouse wake phase? And of course it wasn't effective. So what's going on now is that the drug is being introduced to humans at the right circadian time, at the beginning of the sleep phase, and it's showing to be effective. So again, we've got to be very careful extrapolating from mouse data to human data and backwards and forwards. Of course, mice are fantastic models, but we need to make the extrapolation time, biological time, relevant. So time of day really matters. Okay, circadian rhythms, sleep, and the impact of sleep and circadian rhythm disruption. So if we think about shift work, extended hours, light and noise pollution disrupting sleep, illness, stress, all this stuff feeding in to promote sleep and circadian rhythm disruption. And the effect is to provide physiological disruption and psychosocial disruption. Let me illustrate what I mean. Physiological disruptions, a distortion in the hormonal axis, the hypothalamic pituitary axis, changes in cortisol, thyroxine, reproductive hormones, growth hormones, changing memory and cognition, leptin and ghrelin, the hunger hormones, and this new glymphatic system, which has been discovered and clears out misfolded proteins like beta amyloid, which are associated with Alzheimer's and dementia. Psychosocial stress is you're tired, you're disrupted, you have difficulty coping. That can promote low self-esteem. Social issues, frustration, anger, increased worry, anxiety, and depression. And these two sort of cycles collapse into each other and produce cognitive, emotional, and physiological problems, pathologies. And of course, they can feed back and make the scarred worse. So the key point is that, you know, sleep-wake disruption is not just feeling tired at an inconvenient time, but is associated with both short-term and long-term problems. And so let's look at those issues in a bit more detail. We can look at our emotional responses, our cognitive responses, and our chronic impact upon physiology and health. This is after years, but these can kick in after just a few days. Emotional responses after scarred, fluctuations in mood, irritability, anxiety, loss of empathy. You fail to pick up the social signals from friends, family, colleagues, increased levels of frustration, risk-taking impulsivity, doing stupid and unreflective things. Very nice data showing that the tired brain remembers negative experiences. The tired brain remembers negative experiences and forgets the positive ones. So one's whole worldview is distorted if you have a tired brain. Encourages stimulant use such as caffeine and then reversing the stimulants using sedatives. Cognition. Our whole cognitive performance declines, our ability to multitask. So extracting from the multitude of information coming in, what's important. This falls apart if you're tired. Memory consolidation, and I'll show an example of that. Attention, concentration, communication, decision-making, creativity, productivity, motor performance, and social connectivity. All of the really essential things that make us able to function optimally are massively impacted by scarred. Long-term, as has been shown in night shift nurses, for example, and other night shift workers, you get high levels of daytime sleepiness and microsleeps, no great surprise, cardiovascular disease, altered stress responses, high levels of cortisol, for example. They suppress the immune system, which is probably associated with the higher rates of cancer you get in night shift workers. It's worth bearing in mind that the World Health Organization has now classified night shift work as a probable carcinogen because of the higher rates of cancer you get in night shift workers, metabolic problems, depression, psychosis, and even dementia. So this is really big time health issues. We discussed these in great detail in Lifetime. This is the UK version, and I show the American version because actually most people prefer the American cover, and I let me know what your thoughts are. Yes, it's interesting. So anyway, all that stuff about scarred and sleep-wake disruption is detailed in great in the book. I just want to show you a couple of examples: scarred and memory and cognition. This is a wonderful study by Jan Born in Germany, and he gave people a cognitive task. It was difficult to do. And so he gave one group the task in the morning, and they had to, so they familiarized themselves in the morning and then did the testing in the afternoon. And you see that 20% of the group solved the problem. The next group were introduced to the task in the morning, but they performed it the following afternoon, but they got no sleep. And interestingly, they were no worse than the individuals who got the task in the morning and had to perform it in the afternoon. Of course, the interesting group are those that got the task in the morning, exposure to the task in the morning, they familiarized, then they performed it the next afternoon, but with a normal night of sleep. And you see that 60 to 70% of the group solved the problem. Brilliant illustration of the importance of sleep for problem solving. So sleep promotes the ability to come up with novel solutions to complex problems. Mental illness. The majority of individuals with a diagnosis of mental health problems experience some form of clinically significant sleep disruption. This has been known since Kraepelin, the father of psychiatry, way back in the 1880s. Hasn't been looked at quantitatively. So we decided to look at the impact of schizophrenia on sleep-wake biology. Now, what you can do is use these little devices called ActiWatches, which can measure movement. And we see here's the activity of an individual. This is midnight. So they were going to bed after midnight and they were asleep. You can also measure melatonin. So you can be onset, wake. You can also measure melatonin in urine, and you can find out where the peak of melatonin is. So you've got two markers of where the clock is, activity and hormonal profiles, and you can combine this with a whole bunch of questionnaires. And we'll touch on this right at the end of the presentation. These are the patterns that we found. This is the graph I just showed you from an unemployed individual, and this is somebody who's active way after midnight. Their melatonin is a bit wobbly. This is an individual who's drifting through time. Here's somebody who was drifting, they kind of hung on. Their melatonin was drifting completely the wrong time. Then they fell apart here. Massive all the way through to this person with no 24-hour. These rhythms weren't just bad. They were the worst rhythms I've seen in anything. They were utterly smashed. And that made us think of what could be going on, not only in schizophrenia, but in other mental health conditions. And we decided to think about the relationships between sleep and circadian rhythm disruption in mental illness. And we asked the question, what happens if you stabilize SCARD in mental illness? And I won't go into the details, but this is a study I did with my colleague Dan Freeman in psychiatry. And these are individuals, young individuals, showing paranoia and hallucinatory experiences, and they had poor sleep. This is the control group. So this is their level of paranoia. I'm just showing paranoia. Before and after, there was no intervention here. There was no attempt to stabilize sleep. So basically, the levels of paranoia were the same, as were the levels of sleep, poor sleep before and after. But in the sleep intervention group, where sleep was poor before, but then improved, you see a very significant drop in levels of paranoia, and indeed with hallucinatory experiences, showing that sleep stabilization can be an important therapeutic intervention in circadian rhythms. In mental illness. Okay, so let's now move on to really the last part of the talk, which is the importance of DNA methylation, circadian rhythms, and age, and how this can feed into future approaches for developing drugs, but also our trial and how the trial is going to be really informative in some of the fundamental questions that we have. So DNA methylation, and many of you won't be familiar with this, so I've gone through with a few bullet points. DNA methylation is an epigenetic mechanism involved in the transfer of a methyl group onto the carbon 5 position of cytosine in DNA to form a 5-methyl cytosine. Why is that important? Well, DNA methylation regulates gene expression by recruiting proteins to the DNA involved in gene regulation, or by inhibiting the binding of transcriptional factors to DNA. During development, the pattern of DNA methylation in the genome changes as a result of dynamic processes involving both the new methylation of DNA and demethylation of old methylation sites. And then finally, and this is important, multiple physiological environmental factors can elicit changes in the DNA methylation pattern. So the environment can change the methylation of DNA, which can in turn change the expression of genes. And this is just an illustration of that. So what we've got here are physiological environmental factors which are driving a methylation of cytosine. And of course, you can have the reverse of that with the demethylation of cytosine. And as I say, this is an important site for the regulation of DNA. DNA methylation and aging. Now, what's important is epigenetic patterns change over the lifespan. We don't have the same methylation pattern as we age. Age seems to cause characteristic DNA methylational changes that underpin normal aging and may be precursors to disease. We don't fully understand what's going on here. We want to get a better idea of this. Such changes form the basis of an epigenetic clock, which translates the genomic methylation profile, how the DNA has been methylated, into an epigenetic age. So you can look at the level of methylation of key genes and get a sense of how old somebody is. I'll illustrate this more in a moment. Epigenetic age estimates both chronological age, how old you actually are, but can also predict the risk of chronic diseases and mortality. And then finally, defining which environmental and physiological factors impact upon epigenetic age remains a fundamental goal in the biology of aging and is something we'll address in the Time ZZZ trial. And this illustrates what I've just said. So, for example, from birth to death, you have changes in DNA methylation of cytosine, and we know that risk factors such as smoking and alcohol, and perhaps, there's limited data at the moment, and that's what we're going to explore, scarred and promoting biological aging. Whereas you can slow aging and change the methylation profile by healthy environmental interactions. Okay. The key point is that physiological environmental factors can affect DNA methylation, but can also affect circadian rhythms. So we're in a very interesting situation where DNA methylation can regulate the expression of clock genes, such as clock and BMAL, and I'll talk about those very shortly. And so methylation, DNA methylation, can change the molecular clockwork or the expression of the genes associated with the molecular clockwork. But also circadian rhythms can regulate when DNA methylation occurs. And there's some very interesting data emerging, and this is what we want to build on, showing that circadian dysregulation of methylation may increase your chances of disease. So, for example, shift work in this study here showed changes in the pattern of DNA methylation and increased epigenetic age, which is really fascinating. But as I say, it's early stages, and this is what we want to build on. Okay, changing rhythms with circadian age. We know that there are distinct changes in the circadian pattern, but we don't know what causes those changes. It could be DNA methylation, but we don't know yet. One really important area is the amplitude of the circadian oscillation. So here we have a young individual, high oscillations, very robust circadian changes. But as we age, that amplitude gets smaller and smaller. And when you're near death, you're showing almost no circadian oscillations at all. Now, that's really important because this change in the amplitude of the circadian drive changes masses of our circadian physiology, from kidney regulation and when you produce urine, through metabolism, and I'll show you an example of this in a moment, all the way through to sleep. So this change in circadian drive could be very important. And I've got some data that I'll show you in a moment that can link that amplitude to DNA methylation. Also, another factor which we don't fully understand is that young adults have more sensitive clocks than healthy older adults. There's a drop in sensitivity. If you drop sensitivity, it's more difficult to regulate the clock, and therefore sleep-wake cycles and other areas of physiology begin to fall apart. We have no idea what's causing that drop in sensitivity. It also occurs in mice. So here's a young mouse, and we've turned on this gene called FOS. We see lots of light-induced FOS in a young animal in its biological clock, but an animal that's older, it's dropped significantly. So the signal, the light signal to the clock, as well as the clock itself, is changing as we age. We don't know what's causing that. Okay, I will skip this bit because of time, and just want to go to some of the work that we're doing on our future approaches and translation. So have a quick sip of water. And I will go through this fairly quickly because the details remain sort of interesting, but are not fundamental. So I've talked about the molecular clockwork, but I haven't gone into details. And so here's our single SCN neuron, and we take away the face of the clock. And what we see are two key genes, the PER1 and PER2 genes, and the Cry1 and the Cry2 genes. And they have a regulatory element called an EBOX. Now what happens is that clock and BML bind that regulatory domain of the gene. They drive transcription, translation into the proteins. Those proteins form a complex. They move into the nucleus and they turn off that BML and clock transcriptional drive. So no more drive, but the proteins are then degraded. And then clock and BML can be turned on and the whole cycle can repeat. So you get this beautiful, excuse me, molecular oscillation I talked about earlier. Well, that's the clock, but that doesn't explain how it's regulated by light. And for that, we need to look at the PER1 and the PER2 genes and these regulatory domains, AP1 and CRE. And what I've been doing over the past few years with my wonderful colleagues, Arti and Shri, is trying to work out how light regulates the clock. And I'm just going to give you a brief overview. Okay, so, and I took a long time animating these, so I hope you appreciate it. Here's our photosensitive retinal ganglion cell. It sees light. It sends down action potentials, electrical signals to its terminals. They cause the release of neurotransmitters, glutamate and PACAP, and that increases the levels of these signaling molecules, cyclic AMP and calcium within the neuron. Cyclic AMP controls a kinase, which then phosphorylates CREB. Calcium also changes the activity of a kinase, but in this case, it dephosphorylates CRTC1. And the dephosphorylated state, it can join CREB to find a partner. That binds to this regulatory domain on the DNA, on the PER1, PER2, and you get the production of PER1 and PER2, and the production of those proteins, PER1 and PER2. But it's more than that. There's also a CRE domain on the gene CFOS. CFOS has a binding partner called June, and when these two are working together, you activate this other regulatory domain and have a second wave of PER1, PER2 transcription. And the net result is these genes are turned on, slightly different rates. Probably PER1 and PER2 have different AP1 and CRE domains. But a very important point is, yeah, sure, this explains how light turns them on, but what turns them off? And we discovered that there's yet another thing going on, a gene called SIC1, which is activated. It's also got a CRE domain here, here, here. It turns on SIC1, is a protein. It then rephosphorylates CRTC1, and in the phosphorylated state, it can't bind to its partner. So this whole thing collapses. Everything's turned off. Take away the light, and then everything goes back to the resting state. So it's really exciting. We have an exquisite understanding of how light is regulating the clock. Okay, I'm just going to show you a bit of data on SIC1 because it's so cool. Bear with me on this one. This is the cartoon of what we did. And so what we're going to do, here's our light-dark cycle, and this is when the mouse was active in its running wheel. We're going to shift the light-dark cycle, and you see that the mouse takes about 6 days to adjust to a 6-hour shift. But we've now taken away SIC1, that break on the effects of light on the clock. And it's just so cool because here's the light-dark cycle. Here's the entrained mouse. We advance the light-dark cycle by 6 hours. And it doesn't take 6 days, but it's adapted within a day. It's remarkable. Those are the cartoons. Here's the real data showing the control drifting through time. Here's the very rapid readjustment. So what we now know is that SIC1 acts as a brake on the effects of light on the clock. It makes light less effective. And SIC1 is why we get jet lag. And some of you may be interested to know a bit more about SIC1, and we can deal with that in the Q&A. Okay, translating these findings. The problem, of course, is that sleep and circadian rhythm disruption comes in a variety of forms. We've got a drifting clock, which is found in individuals, mental illness, no eyes. You saw that drifting clock of that individual with schizophrenia. No eyes are the same. Neurodegenerative diseases, insomnia, dementia, neurodevelopmental disorders, mental illness, and of course, completely fragmented non-24 hour. And again, you saw an example of that with one of our individuals with schizophrenia. So what can we do about these conditions when behavioral change is either ineffective or not possible? So we've taken some discovery approaches to correct sleep and circadian rhythm disruption. And we've done both pathway analysis and drug repurposing. Let me explain. So what we can do is we can take cells and we can attach a reporter to these clock genes. And the bottom line is you can see these genes being turned on and off, and this is speeded up over a 24-hour day. So we can look at clocks in a dish and measure their circadian rhythms. We can also do that for the SCN. So here's a single SCN, and you see it's turning off and on over the 24-hour day. And of course, we can look at behavior, which means we can look at the effects of drugs and compare a normal oscillation with a drug that's shifted the oscillation in time. It's delayed the circadian rhythm, and another drug which increases the amplitude of the circadian rhythm. And I'll just show you a couple of examples. So example one is essentially a drug that fools the circadian system that it's seen light. So it's promoting an increase in cyclic AMP and calcium, which is triggering that whole axis that I talked about. Again, a cartoon to show you what we've done. Here's a mouse in complete darkness. So its clock is drifting through time. We're now going to introduce the drug, and we see what happens is that the clock and the behavior latches onto the drug just like light. Take the drug away, and it drifts through time. And there's the cartoon. This is the real data. You can see no drug here. The clock drifts through time, but the exciting data is that here's an animal in complete darkness. You introduce the drug here, the activity locks on. So it's, again, just like light. Take the drug away, and it drifts away. So we've got a pharmacological replacement for light. That's going to be immensely powerful for people with no eyes, profound blindness, and in many psychiatric conditions. So we finished the early animal stuff. We've done the first in human trials showing it's safe, no problems, and we're now ready to do the big trials to get this into the clinic. The second example is a drug screen, and this is not part of the pathway analysis. We weren't expecting to find this. We were just screening drugs, and we found a drug that increased the amplitude of the circadian rhythm. This is early data, and I just want to give you a sense of what increasing the amplitude of the clock can do on physiology. And in this case, it's metabolism. So we're looking, interestingly, in an epigenetic modifier, a drug that changes methylation of the genes PER1 and PER2. And what we can do is look at, and as I say, there's an intimate relationship between circadian regulation and metabolic function. So here's a mouse that's been fed on a high-fat diet, and you see it has high levels of fatty liver. And of course, fatty liver disease is a phenomenon in humans. This high-fat diet is then compared to a mouse here, which is given a low-fat diet, and we see no fatty liver disease. Okay, what happens if we take a mouse that's been fed on a high-fat diet and given this drug, which increases the amplitude of the circadian oscillation by changing DNA methylation? You completely reverse the fatty liver. Both we looked at 10 weeks and at 18 weeks. So just by changing the clock, you can have a big impact upon metabolism in mice. Applications, of course, huge. We talked about the diminishing amplitude of the circadian system in adults, in dementia, neurodevelopmental, and all the rest of it. And so by increasing the amplitude of the circadian drive, we can perhaps increase the stability of physiology across many areas of health. Finally, let me just introduce the Time ZZZ trial. Now, this is started, and let me give you some background. So Time ZZZ is Elysium's new longevity study focusing on sleep, and it's registered as a proper trial. Our goal is to advance our understanding of the association between chronotype, which we talked about, whether you're a morning type, an evening type, the quality of the sleep that you get, and associated with that, all the problems that you get with poor sleep, mental health, for example, and biological aging. And both Dale and I are the investigators on this project. We're collecting data, first of all, by questionnaires. So we can collect details about sleep, chronotype, anxiety and depression, and diet, and general information. So this is gentle questionnaire data. And in a subset of individuals, we'll also look at epigenetic data and get a sense of the epigenetic clock that we talked about earlier. What's the pattern of methylation in these individuals with these different conditions and different phenotypes? And we can get wearable data by essentially, as I showed you earlier with schizophrenia, measuring rest activity cycles. Our primary endpoint will be determine whether individuals working out of phase with their chronotype exhibit higher rates of biological aging compared to those working within their chronotype. So fundamental information that we have limited information about. Exploratory endpoint 1, investigate the relationship between chronotype, alignment, and depression risks in relation to the rate of biological aging. Exploratory endpoint 2, identification of methylation markers associated with chronotypes. So as I said right at the beginning of this talk, we know there are gene changes associated with being morning or evening, but are there epigenetic methylation markers associated with chronotype? I think there almost certainly will be. And then finally, endpoint 3, identification of epigenetic markers associated with depression. Can we get some idea of individuals who are depressed or showing borderline depression? Will they have key methylation of their DNA in specific genes? Can we get a better marker for the epigenetic association with depression? So key goals. Anybody is eligible to take part if you've taken the index test registered through Elysium. So this test here, and you can find more about this on the website. What does participation mean? Well, first of all, it entails just the questionnaires. And then we'll go on and in a subset of individuals, look at the more detailed analysis, should you be interested in taking part in that. And I do hope you would be. And then, oh yes, for the first 100 to complete the questionnaire, we've given a copy of my book, Lifetime. And now I heard from Dale earlier that we've already got 180 people signed up. So the 100 copies that are going out to those people who've signed up. Really above and beyond the book, which I'm very delighted that Elysium has decided to give to participants. The individuals will get real insights and contribute to advancing clinically validated interventions and health for monitoring tools for longevity. So it's great to be part of such a study. And finally, because I realize I've overrun, if you want to know more about Time ZZZ, here's the little matrix that you can scan into your phone, and that will take you directly to the website. So with that, I apologize for overrunning. We did cover a lot of ground, and thank you very much for your attention. So Dale, back to you.
Thank you, Professor Foster. Thanks for sharing your insights and expertise with us today. I think what struck me through that presentation was the incredible depth of knowledge that you have, and it was truly a pleasure to hear you speak. A couple of interesting pieces that I pulled from it. As a father, I'd never actually considered the impact of chronotype or circadian rhythm on optimizing learning outcomes for kids. So that was a unique insight.
I think that, shifting exams to the early afternoon, you can improve exam success, and that's when the kids are more alert. It's really remarkable.
It makes perfect sense to me, and I'd never actually considered it before. But I think more importantly, and you highlighted this very well in your presentation, is the importance of sleep and how it just cannot be overstated, its impact on biology, which is why we see all-cause mortality and sleep so heavily linked together.
Yeah, I think it's also extraordinary that sleep is poor sleep is so much more than feeling tired at an inconvenient time. It has global impacts upon our cognitive, emotional, and our physiological health. And it's extraordinary to me, why more attention isn't being paid to this.
Yeah, absolutely. So just a couple of closing remarks. Thanks again, Russell. To all of the attendees, thank you so much for joining us, and thank you to those who have submitted questions already. This webinar recording will be sent out to everyone through a newsletter, and there'll be also another opportunity for those to reply with additional questions. And our plan is to go through those questions, consolidate them, and send back responses in another newsletter. We hope that today's discussion was enlightening and informative to everyone, and I wish you all a wonderful day. Thanks again for jumping into today's webinar.
Thanks, Dale, and I'm really looking forward to working with you on the project.
Me too, Russell. Thank you.
Time ZZZ.
Yeah.
All right. Take care, everybody. Thanks for listening.
Bye.